Okay, so we're going to move on now to our next presenter, which is Greg Fahy from Intervene Immune. Is Greg with us?
Well, I wanted to thank the organizers for allowing me to have a chance to explain to everybody how Intervene Immune is hoping to spearhead the immune and epigenetic frontier in human aging intervention from sunny Southern California. So we started with the premise that immune system aging is a universal and very often fatal aspect of aging that is programmed in youth. As you can see from the diagram, you start off as a newborn with a lot of functional mass in your thymus, but even by the time you get to be a teenager, the thymus is gradually being replaced with fatty tissue. By the time you get to be about 40 or 50 years of age, there's very little functional mass left in your thymus. Unfortunately, that's really important because the thymus manufactures your T cells. And although T cells last for a long time, and although there's still a trickle of T cells coming out of the thymus at all ages, the supply of T cells diminishes over time. And as a result of that, somewhere between the ages of 62 and 78, you lose about 98% of your ability to recognize foreign antigens. And unfortunately, that also has significant clinical effects. So there was one study looking at immune system competence in 80-plus-year-olds. And as you can see, the arrow of aging drives you up this death risk curve from less than 40% to 80% risk of being death in 2 years in this particular case. And unfortunately, the probability of being in the low immune function group was higher than the probability of being in the higher immune function group. So this affects large numbers of people, and it's probably a leading cause of death. Now, we've all known that 90% of all of the flu deaths that take place are in the people who are 65 years of age or older because of immune incompetence. And pulmonary disease and death is a problem in this group as well. But of course, the COVID-19 crisis has really highlighted the problem with having a poor immune system as we get older. Why is it not advancing? Uh-oh, it's not advancing. I don't know why.
Okay, well, are you driving from your end, Greg?
Oh, yeah, okay, finally, yeah. I think it looks like it's working again. Okay, I don't know why it didn't. Okay.
Great.
Sorry about that. Okay, so one other feature here is that there's a lot of very exciting interventions to deal with aging in general that are out there, and I've listed a few of my favorites on the top of the slide, but we believe that these approaches are not going to reverse immunosenescence because thymic involution is programmed in youth. It's a biologically controlled process, and it's not really part of what you think of as normal aging. For example, heterochronic parabiosis does not reverse thymic involution, although it reverses many other aspects of aging. Other approaches have been tried to bolster the failing immune system that we already have, but they really ignore the main problem, which is the lack of functional T cells. So we showed last year in our paper, An Aging Cell, we reported on the results of our first trial, the TRIIM trial, that immunosenescence actually can be addressed today, apparently. So what you see in the upper left is MRI images of the thymus before and after treatment. You can see the whitish, fat-laden thymus reverting to a darker thymus, which is enriched in functional T cells. You can see the quantitative results to the right of those pictures with very high statistical significance, both for thymus and also for bone marrow fat regression as well. It was interesting that as the thymus got more gray, a lot of people's hair got more gray as well, turning from white to gray, and up to 3 out of the 9 volunteers, something we were not really expecting to see. As far as the things we were expecting to see, our immune parameters behaved the way we wanted them to. We saw an increase in naive CD4 and CD8 T cells. We saw an increase in recent thymic immigrants. We also saw a reduction in monocytes, which is significant, and we'll go into more detail about that in a second. And we also saw improvement in a variety of different kinds of cancer risk factors. For example, we saw a reduction in exhausted CD8 T cells bearing PD-1 as a marker. PD-1 is a checkpoint inhibitor that blocks the rejection of cancer as we get older. We also saw that markers for prostate cancer risk improved both PSA went down and percent-free PSA went up. On the other side of the immune coin, we saw that age-related inflammation went down, at least as measured by CRP, and the lymphocyte to monocyte ratio improved, which is correlated with improved outcomes for many different kinds of cancers, as well as for cardiovascular disease and all-cause mortality and stroke. We also saw an improvement in kidney function over time. And the other thing I'll highlight here is that the side effects that we saw were all mild and dose dependent. There are many other details in the paper. I invite you to read it. It's open access, free to anyone. The thing that really caught people's attention, though, was this discovery, which is that our treatment reverses epigenetic aging. This is the first time this has been shown in any animal, let alone humans, but that's what we saw. So we used 4 different epigenetic aging clocks, and they all showed broadly the same thing. And you can see the lumped result over all of the clocks on the extreme right. And what you see is that at the end of 1 year of treatment, the individuals in the trial were about 2.5 years younger than they otherwise would have been. Or they were about 1.5 years younger than they were when they entered the trial, if you factor in the fact that they were 1 year older by the end of the trial. Another interesting feature is that the epigenetic aging reversal that we saw accelerated over time, so that in the last 9 months, the last 3 months of treatment, epigenetic age was going in reverse at the rate of about 6.5 days for every day of treatment. And that was statistically significant. We also saw that there was no correlation between the benefit that we saw epigenetically and the age of the individuals at entry into the trial between 51 and 65. And these results were sufficiently provocative that they received some mention in Nature and Science, and we were very gratified to see that. So what are we going to do next? Well, we first of all have to replicate these results and extend them. So we're going to be launching the TRIIM-X trial probably next month. And it's going to be a phase 2 randomized controlled trial. In the original TRIIM trial, we only had 9 Caucasian men. In this trial, we will have women and older men. We'll also have multiple genetic backgrounds, and these will be randomized across all of the different groups. We'll be using telemedicine, so we'll be accepting enrollees from across the United States, and we hope to be enrolling about 10 to 15 volunteers per month. We already have enough volunteers that have gone through preliminary screening to form the first 2 cohorts. We have about 500 people on the waiting list waiting to get into the trial. The trial will have at least 5 treatment groups and 1 placebo-only group. And we're going to manage the data stream coming from this trial by further refining proprietary computer-assisted data analysis and dose updating program that we've developed, which will allow us to scale later on as well. And we'll include new treatment measures such as immune cell function tests, measures of frailty, et cetera. The trial will proceed in a series of iterative steps in which we draw blood, we look at the status of each individual in the trial, and then we do dose adjustments based on how each person in the trial is doing. It's a very personalized medicine approach. We're on clinicaltrials.gov. If you'd like more details about the trial, you can read about them there or contact us. So we started off around 2015 with the TRIIM trial. It finished in 2017. We filed for patent protection in 2018. We published in 2019. This February, the FDA approved the TRIIM-X trial. We were accepted into our new headquarters in April. In May, we submitted a large grant to help us support the costs of our trial. And since then, we've been improving our software and seeking grant support and seed funding. The launch of the TRIIM trial will probably be, as I said, next month, maybe even at the end of this month, but probably not going to quite manage to do that. And then at the beginning of next year, we'll be talking with the FDA about how we can move forward with clinical services. We have an accomplished team at our disposal. I'm the designer of the TRIIM trial and of the TRIIM-X trial. I wrote a book and edited it called The Future of Aging. I've got several patents and have received several awards for my past research. Robert Brooke is our CEO and CTO. He founded Iovance Biotherapeutics, which is now traded on NASDAQ under the IOVA ticker symbol. He has investment analyst experience with Bristol Capital Advisors. He's currently an advisor to the X Prize Foundation and sits on the board of the Clock Foundation. Paul Hynek is the CFO. He has started his own company, which manufactures software for businesses, but he was also the former director of business affairs at Giant Studios, which made a number of blockbuster movies. Of course, everybody knows Steve Horvath, who's the father of epigenetic clocks for aging, and is, I think, undisputedly the world leader in that field to this day. He's our chief scientific officer, and he's done the epigenetic analysis in the TRIIM trial. He will be further assisting us with TRIIM-X. He's at UCLA, of course, and is the co-founder of the Epigenetic Clock Foundation. Joe Sugar, like Paul Hynek, was a volunteer in the TRIIM-X, in the TRIIM trial. He was the former dean of the Florida State University College of Medicine. He's given many scientific presentations in medicine, received a number of awards and recognitions, has written huge numbers of papers and written several books. And Dr. Shreyas Vasanawala is the chief of body MRI at Stanford University, where he holds a number of similar positions. Now, I don't really have to tell this audience this, but the U.S. market alone in the field of aging intervention is quite large. As it pertains specifically to thymus regeneration for preventative medicine, we figure everybody over the age of 50 is a good candidate for this, at least until we learn something to the contrary. If you figure that you might want to regenerate your thymus once every 10 years, then even if we only cleared about $1,000 on each of these patients, you can project a significant addressable market. We also think that we can parlay thymic regeneration into cures for autoimmune disorders and to prevent transplant rejection. We're also going to be working on next generation pharmaceuticals, including growth hormone biobetters. Of course, the future is hard to project, but we can make some projections based on a variety of scenarios, indicating that we should not have to incur a lot of debt and we should be able to become profitable in the near future. We have several proprietary advantages. We certainly have a unique approach to a very serious unmet medical need. We're definitely the leaders in bringing these particular therapies to people. There's a demonstrated demand. We have compelling statistics in the sense that in the TRIIM trial, we only had 9 individuals, but we had highly statistically significant results, implying that we'll be able to replicate these results with larger populations to the satisfaction of the FDA. And we have several endpoints from our trial that may enable us to create a low bar composite endpoint for FDA approval. Most new drugs have what you call the valley of death, in which everything seems fine until you go into the clinic and then you discover that you're not going to make it through to the other side. But we don't think that we're going to have the same risk because our pharmaceuticals are vanilla, growth hormone DHEA and metformin. Everybody knows about them. The risks are well worked out. The FDA is not concerned about the dangers involved. And we saw no major side effects in our trial, and there's a lot of clinical and animal experience to back that up. We will have proprietary computer algorithms and telemedicine, which will enable us to have a unique ability to scale our treatment across the United States and then to the rest of the world, even though it's a very personalized medicine approach. We already have partnership interests from the United States, UK, Europe, and Asia. And we think our GH biobetter or biosimilar will be very useful for controlling costs and improving the properties of available hormone, as well as having other advantages. And we're beginning to develop institutional alliances, such as with the Epigenetic Clock Development Foundation and the X Prize Foundation. We also have this reservoir of epigenetic age reversal information that's unique to us, which will be greatly expanded by the TRIIM-X trial, which we believe we can parlay into many different insights into how human epigenetic aging reversal works and how we might be able to improve it. We do have an international patent pending, and we expect to have many more as time goes by. So as Alan Kay once said, the best way to predict the future is to create it. That's what we're trying to do. And we invite anyone who's interested in helping us to contact us. And I appreciate the opportunity to speak. Thanks a lot.
Great, Greg, thank you. We have a few questions that have come from the floor. So first one is, personalized dosing scheme would require a high workload of monitoring and dosing adjustments. Would this be a big burden to the patients and healthcare system?
It wouldn't be a burden to the patients particularly. We'll be able to streamline that to some extent going forward as we learn much more about how this works. But everybody's an individual. Certainly in the TRIIM trial, we had 9 guys in the trial, and we had 9 different kinds of responses. But there's ways of managing that complexity. So we think that the computer model of computerized decision facilitation will be critical to scaling this to a large market. Clinician partners only have to assign blood draws based on the feedback that we require. The patients so far have not had a problem getting their blood drawn fairly often. And as I say, we'll probably be able to reduce the number of blood draws as we go forward.
Okay, and what treatments are you considering for TRIIM-X?
Right, so for TRIIM-X, we'll have 6 treatments. One will be a replication of the TRIIM group, but with a broader genetic background. Another will be for women of the same age range, and another will be for older men, older than 65, between 65 and 80. Then we're going to have 2 other kinds of control groups, which have all of the other features of the full treatment, but lack growth hormone. Because some people think that you don't need growth hormone to have epigenetic aging reversal. We're pretty certain that you need to have growth hormone to reverse immunological decline, because the other agents we don't think have any effect on the thymus. But if you're not interested in that, if you're mostly interested in epigenetic aging, then it may be interesting to see what happens without growth hormone. And in fact, there's a profound question, based on biology, as to whether growth hormone, which people usually consider to be pro-aging, might be anti-aging in our context. So we'll find out about that in this next trial.
Very good. Well, Greg, you're bang on time. So I guess you're going to get some further questioning in the breakout room. So thanks for a great presentation today. Really, really appreciate it.
Thank you much.